Then we can also access the databases that are online, such as NCBI, Gene Bank, and also Uniprot.
Yep.
We will use some bioinformatic tools that are commonly available, and also some other tools that will be shared by the different lecturers in this course in the coming weeks. And one of them will be also on structural bioinformatics, where you learn about the protein structure. And because our course is just one small topic, there's no coding involved, but there will be some use of Linux in the following weeks, I guess next week. So bioinformatics is a very big field, and it involves many people, as I mentioned to you before, not just biologists like us mainly, or life scientists, but also engineers and computer programmers who are not biologists, but they will do all the work that allows bioinformatics to take place. And also now we're beginning to see the use of AI, or artificial intelligence and machine learning, because the data is already too much even for bioinformatics programs. Even though they are bioinformatics programs, it is still a human that run the codes to analyze the data. But that is becoming impossible now. So in order for us to understand where we are today, I will share with you a story about how scientific progress came to be, and also that all along we never knew what we were doing, and we still do not know what we are doing. Okay? So this is the story of humanity, because we learn by trial and error, right? So it doesn't mean that we are to blame, but certainly we are blind in terms of present, in terms of the present times that we live in, because we are aware, but we do not know what are the wrongs or rights. Example that everybody can know is that how does the earliest humans know which mushrooms are poisonous?
Right.
Yeah, so it will be somebody eat and there is some bad effect, right? So then they learn over time, many generations. Mostly groups of people, so they live mostly in close to the jungle, and even though they do not have formal education, but they are also tourist guides, because they are guides and they can tell you which plants to eat, which plants are good for certain medical properties, even though they do not have formal education. So that is the kind of knowledge that we learn from trial and error, and by passing on that knowledge. So this story starts in London in 1800s. So this is 1852. Okay. So in 1852, if we recall history, especially for Malaysians and also other people in other parts of the world, you will know that the British Empire is already quite widespread, right? So they've gone to India, they've gone to Nepal, in the Middle East, even in Africa. They are still very rudimentary, in the sense that because London is a city, a lot of people will go to the city because that's where the economic activities are, and they will bring with them livestock like pigs and sheep and cows, and there are records of that they will actually put their cows in the where together with where they live, which means that if they live on the third floor of the building, they will also bring their cows up, and they do not have a sanitation system. By our own waste and also animal waste. in London in 1800s, there are three main outbreaks. And you see here, they are diphtheria, scrofula, and cholera. But have you heard of some of these diseases before?
Yes.
Yeah, I think everybody has heard of cholera, right? So even though we are aware of this today, because all of us have gone through some kind of formal education, in unclean drinking water. But back then, nobody believed that these diseases are caused by dirty water or unclean water. So the theory at the time is that because of the pollution in the city of London, all these diseases are caused by the unclean air. So the prevailing theory in 1800s is that it is the dirty air that is causing the spread of this disease. Okay? And nobody believes that even though they are living with animals and they are discarding their waste into the streets because there is no sewage system, nobody believes that it is the water that is the cause of this. So it is only until there is this doctor in one particular area in London, and to understand this map, so one bar indicates that there is a death in this particular building here. So along this street there is one drinking water pump. So during that time there is no tap water, you can't just turn on the tap at home, but there is a common pump, and also this is still maybe bacteria. So he was the first. Everybody that is drinking from it is dying, especially those who are living near to the pumps. But of course this doesn't catch on. People So this is one of the first cases of epidemiology where you show that there's a link between the causative agent and the spread of disease. And at the same time, in another part of the world, that individual that has the disease. Yeah, this time is in Italy. So although Italy is very close to the UK because they are in Europe, but still there is no scientific communication, and whatever that was found out by John Snow doesn't necessarily get spread to other parts of the world. So he also independently discovered that there is something in the water or in the patients, in the body of the dead patient, and that microorganism is called Vibrio because it's shaped like a comma. Maybe like 30 years later, after the 1850s, so now we have some kind of more understanding about microorganisms and bacteria. And if you have studied microbiology in your undergraduate, then most likely you have heard of Koch's postulates, right? So Koch's postulate is very interesting because for the first time they show that by using mouse, okay, you can infect the mouse with this particular suspected pathogen and then isolate and you use this isolate to infect again one more round of the mouse, just to confirm that it is truly in fact that bacteria. Okay, so it doesn't stop at this stage only, but by confirming that you can reinfect the model organism, so it will show that this is actually caused by this same pathogen. Okay, so this is sort of the start of, not maybe the start, but one of the examples of the scientific process where you have to show proof, right? You claim that okay, can without a doubt cause this disease. And although he won the Nobel Prize later on, it is interesting that one of his also assistants that studied Salmonella typhi, which caused the typhoid fever. Okay, so have you heard of Salmonella typhi? Organism that spread through food or water. So Salmonella typhi doesn't have this particular model. Do you have any idea why? So why do you think Salmonella typhi, bacteria, gram-negative bacteria, it cannot infect a mouse?
Because it doesn't have the receptor places where it can.
Yeah, so it doesn't have a receptor, and also because Salmonella typhi is a human-restricted pathogen. So it can only be spread from humans, so the host is only humans. There is no intermediate hosts in other organisms or other animals. So unfortunately, because if you read some of the stories or biographies about this, then it's kind of sad. So it's like, although he's not wrong because he's doing this Cox postulate by testing on mouse, but he couldn't show the proof that this bacteria is actually killing the mouse, because he can't, because that typhoid is only affecting humans. So bacterial culture and microbiology has developed very much in the 1900s until today, where we have more and more systematic approaches to culturing bacteria and also to test for different things like antimicrobial resistance. And we understand, of course, very clearly about where cholera comes from and how we can use microbiology techniques to do all these things. And much more recently, we have, in the last about 30 years, we have rapid test kits that can be used to diagnose various types of diseases much more quickly in the lab. Okay? So the development of microbiology technologies or techniques also progresses. And that leads us to where we are today. So from many years ago, we have the first discovery or invention of the microscope, which allows us to see that there are actually microorganisms that exist in the micrometer scale. And by knowing that knowledge, we can now develop different techniques such as microbial isolation or media. We can do DNA sequencing, PCR, polymerase chain reaction, to understand all these different microbes. And today, we have bioinformatics to help us, because we have more and more sequencing data that we can no longer analyze or use the old techniques to solve those kind of problems. So the case study that I would like to share with you today is about the use of metagenomics, okay, NGS, because that is still the common term, even though the current one is massively parallel sequencing, and to use it to diagnose a mysterious infection. Okay, this is the story about this boy at the time called Joshua Osborne. So Joshua Osborne has a history of 3 hospitalizations in 4 months, and he spent more than 1 month in ICU. So that's a very serious disease, right? Oops. So that is a very serious disease, and even though the hospital have used more than 100 tests, including rapid tests, PCR, whatever biochemical tests available in the hospital, they could not identify what is the actual cause of the disease. Therefore, because his condition has worsened, he has to be put into a coma. So if you remember the COVID times, it was very serious that some individuals that have a very, bad condition due to their lungs has collapsed and all that, they cannot function anymore. In order to save them, they will be put into a induced coma so that their body is sort of slowed down, right? Rather than have a big reaction to the infection that was going on. So he was put into an induced coma, and also a brain biopsy was taken. So this is the sort of timeline about his story. Okay. He had a trip to Puerto Rico in Southern America in 2012. He started to complain about some symptoms a few months later, August, September, about 1 month later. However, that fever subsided on its own, and he was not hospitalized yet. Okay, so he was still going about his life normally. However, he had some eye infection, which is uveitis, and in the following year, he had another trip to Florida, and that is when he started to get serious. His infection started to become serious. So he was hospitalized for the first time here, and hospitalized again the second time. However, he was still able to recover, but after about 6 months later from these hospitalizations, now his condition has deteriorated. And for if we look at the chart, okay, from this hospitalization period, this is at first the temperature whereby he was admitted on the 7th of October. His temperature has increased above normal levels, indicating there is a fever, right? Yeah. So that is the first kind of sign of an infection inside our body. So they started some kind of medical treatment here. However, his condition became worse. Okay. Then there is some MRI done. There is a brain biopsy. Okay. Altered mental status. I guess it is because he has become weakened and his fever is maybe prolonged. So he now have altered mental state.
What's altered mental state?
Means that you sometimes, if you have fever and you're sick, you become hallucinatory. You could be no longer yourself.
Yeah, schizophrenia.
Not really schizophrenia, but maybe not yourself. Yeah. Schizophrenia is like a different thing whereby you can have different symptoms or multiple personalities, right? So you have schizo, which is a separation of the personality. And so this one indicates the hours. Let me see. So 7. So by this state, because that he is very serious already, they put him into a coma. So this particular yellow region. Okay. And because they have run out of tests to be conducted on him, nobody knows what is the cause of this infection, right? So there is a guy called Dr. Charles Chiu. So Dr. Charles Chiu is a specialist in bioinformatics or genomic sequencing, and he is actually located in another part of America. So because the doctors, one of them heard about this new technology. So remember, this is 2012, right? 2012, 2013. So because metagenomic sequencing is not so common yet, still it is very new, especially in hospital settings. So they say that because there is no more options to run, no more tests that can be carried out, so they decided to try to send his sample to bioinformatics or the next generation sequencing lab to find out what is going on. So they did deep sequencing of the serum and cerebral spinal fluids. So cerebral spinal fluid is usually, if you work in hospital before, it's from the spine, right? They will tap the spine to find out, if, especially for meningitis, where the bacteria hides in the brain. So only from spinal fluid you can detect or diagnose that disease. So with deep sequencing, in about 48 hours, they were able to find out what was the bacteria that caused this disease, and he was treated immediately with penicillin G and also some antibiotics like vancomycin. So by giving penicillin G, his condition immediately resolved or improved because now the fever has dropped, and then other blood count also has gone down, so become normal back again. Yeah. And the reason why penicillin G is the one that is helping and was not given initially is because penicillin G is no longer used as the first line antibiotics, because it is most of the infections or bacteria have now multiple drug resistance. Therefore, doctors or hospitals do not usually prescribe penicillin G at first. Okay, so what happens in detail is something like this. So during this stage, okay, during the days of hospitalization, at this particular dark yellow bar, we can see that after they have sent the sample to the bioinformatics specialist, they will perform the sequencing processing. So sequencing processing involves first the wet lab, where you have to extract the DNA from the sample and then amplify the sample, because the sample is a very small quantity, right? The DNA, so you have to amplify it, usually by PCR, and we call that a library, because you want to make a library for sequencing. And then for sequencing, usually it's just about 12 hours process or 1 day process, whereby you actually sequence to get the DNA sequences, and only 1.5 hours to perform a blast. So blast is just like what you did last week, okay? Whereby you blast the sequences of unknown bacteria against the database, except that this time the blast is for here 10 million reads. The last week you only blast like one sequence, right? So this time you have 10 million reads.
10 million times.
Yeah, so they need to blast 10 million times. Therefore, it's impossible, of course, even if you get many people to blast it. And it also depends on the server, like we have said, if the Wi-Fi is slow.
Yeah.
Do too many blasts.
Trying to blast ourselves then slow.
Yeah.
So G is
Okay.
horrible.
So that means that the entire process takes about 2 days, about 48 hours. And most of it is actually the library preparation, which is to prepare the DNA for sequencing. So after the sequencing, after the blast, they found out that, make sure the speaker, Kavita, you can hear us, right? Okay. Maybe she's doing something. So,
Yeah, I can hear that.
yeah, you can hear. Okay, so that's fine. So after the blast, so they found that there is actually a Leptospira box Petersoni cerebral genome. So they found this strain or this sequence in one of the millions of reads, okay, inside the DNA sequencing. Because the NGS or DNA sequencing is basically sequencing whatever that is inside the sample, right? It's not sequencing one single thing, but it is sequencing whatever that's inside the sample. And apart from that, they do not really find any other things like viral or bacteriophage and all that. So after they have one leptospira found, okay, and they could match it to, so I think this is the one that they found, and then they matched to others. Okay. So how to do this bioinformatically? Okay. So that is how, that is basically the rough process. But in order to do it bioinformatically, you need a computer software, right, to do that. So after sequencing, you have those small reads, the millions of reads. This needs to be processed. Process means that the sequencing reaction will give certain fragments of sequence, and because of the process of generating the sequences, there are some parts which are not the actual results because they are the primers of the PCR reaction. So if you have learned a little bit about PCR, right? PCR, you need a primer. So the primer is a kind of sequence that's needed to generate the library. So the primer sequence needs to be removed, so there is pre-processing. And then remember that this sample comes from a human, so you need to remove the human sequences from before you blast. If not, then you will get just human results. Right? So you need to remove the human sequences. So therefore, by removing human sequences, then you are left with viral and bacterial sequences in the sample. So with this, you can now go to BLAST and perform a comparison with the database to find whether what organisms are found in the BLAST. So these are the details, and we, at our level, we do not usually deal with this much information or do the processing unless you specialize in bioinformatics. And further learn this maybe during your PhD if you want to explore bioinformatics. So the process takes a lot of time, as mentioned. If you were to blast manually, okay, doing a standard blast, needs at least 20 hours or up to 2 years for a complete blast. So there are fast blasts, and also there is the complete blast. Right? So there's the smart blast where they just do a quick search of what's available in the database. So using the sequencing results of more than 2 million reads, they calculated that using conventional manual blast will take about 2 years, or maybe as fast as 20 hours if they cut down on what to search and what not to search. And these are various different programs that are used to blast. So by using a certain method which that particular lab has developed, they were able to cut down the time, and they could get the results in 1.5 hours or even less, the blast results. Okay, so they call this the SNAP method, which is from the previous processing and all that. So this story comes from this guy, Dr. Charles Chu. You can watch his TED Talk on this.
Charles Chu.
Yeah, you can watch. He has a very nice TED Talk. So this was reported in the New England Journal of Medicine. Okay, actionable diagnosis of neural leptospirosis by next generation sequencing. So that means that by having now this technology, okay, compared to Cox postulate and microbial isolation in a lot of pathology lab and so on. So we have next generation sequencing for massively parallel sequencing, which can be used to sequence entire samples, okay, and find what is inside there. So it is very much like just going to the jungle and catching all the animals and find out which animal is the dangerous ones. Maybe it's the tiger or the snakes and so on. So it's something like that. Because
so M stands for metagenesis.
yeah, meta. It stands for meta in this particular case. Meta simply means that everything. Yeah. So in this today there are fields of research called metagenomic research, right? So it means everything. So usually it involves environmental samples where you do not know what you are sequencing or you do not know what you are sampling, but you just capture everything, and then only after that you will sequence and find out what's inside. So according to general information, a lot of diseases, although we may call them meningitis, which just refers to some kind of disease that affects the brain, which causes fever, but we do not know the actual cause of it. Is it a bacterium or is it a virus? We are not sure. Pneumonia is just infections of the lung, but we do not know the causative agent. So maybe metagenomic or meta-NGS may help to identify all of these diseases. However, the cost will be the issue, right? Okay. So, will the hospitals or will the patients be willing to pay for this? That becomes fairly costly. Nowadays, we have RT-PCR in the hospitals in Penang. Yeah, so it's very fast. So I remember when many years ago when my son was sick with food poisoning. So the test that was taken was a stool test, and then it was collected in the afternoon. And then I was surprised by just like 2 hours later, the doctor or the lab can say that it's a rotavirus. Just like a few hours. So I was also shocked that at that time, I think maybe 6 years ago, like in Penang, we have RT-PCR in the hospitals. But RT-PCR is very specific, which means that you only screen for a panel of common diseases. So if you have unknown diseases which are outside this panel, you will still not find the results.
RT in this case means reverse transcribe.
Yeah, reverse transcribe or
Not real time.
no, transcribe, because they just want to identify that there is a presence of that particular pathogen or virus or bacteria. So the goal is to identify all possible microbes from the single sample source. And this is important because in our cells we have a lot of bacteria, and not all of them can be cultured as well. Right? So even though we have already identified many bacteria, but if we are no longer able to culture them, it means that they cannot grow on a plate. So the traditional microbiology technique is that you must be able to isolate the bacteria. But if you cannot isolate them, does it mean that it doesn't cause a disease? So it's also not true. So if the presence can be detected through their DNA sequence, then maybe we are able to now find or identify more bacteria than we currently know. Another example from a publication is actually a very interesting one. So there's this study in Denmark where they did a kind of study to find if they can detect pathogens from the waste in airplanes. Okay, so this is a very interesting one, whereby they collect the waste from the airplanes, the trucks, from long distance flights. long distance because long distance means that people will most likely go to the toilet in like the 10 hours they are traveling, especially if you are traveling from far away. So if you are one of those passengers that carry a certain pathogen, then can they, from the storage tank of the plane, when it is transferred to the truck, then from the truck you sample and find out if there is a disease, right? So this was before COVID, of course. This was a very long time ago, and the reference is here. This was published in scientific reports. So this is a kind of proof of concept again, that you can use computational power to identify dangerous pathogens. Because they are worried also at that time, I think it was around 2016 or so, that maybe there is some kind of bioterrorism where people can transfer or spread diseases to certain countries, right? So during COVID times, of course, there's also studies where they collected samples from wastewater from apartments, I think, sewage waters and so on, that shows indeed the virus can spread through the sewage system. Another example is when this has of course reported again in one of the papers in 2015 that in sometimes whereby the twins who are discordant, meaning that although they are twins, they have the same genetic background, but their phenotype is different. So one is more obese, right, or could gain more weight, and this has been associated with, now we know, microbiota. So microbiota is what is inside our guts, right? So if they collected the microbiota from the slim individual and the obese individual, they transfer it to the same genetically identical mice. However, this one that is transferred with this microbiota becomes obese. So it means that it's not actually the bacteria, it's not actually the host genes or the native genes, right? But it's actually the external factor caused by bacteria. So it means that if you are maybe obese, it is your bacteria that's causing you to eat more or become more hungry all the time.
Wow.
So it's not ourselves.
How do we kill them?
So how can we remove the bacteria?
Yeah, so you have to take the microbiota from the slim person and transfer it to the obese, then it becomes slim again. Yeah. So this is called fecal donation. So in people with IBS is bowel syndrome, right? Yeah, so yes, it is used actually in some clinics or some parts, I think in America, where they have healthy donors, people with no problem, their guts are healthy, and then they collect their fecal matter and they transfer it to the people with IBS, the irritable bowel syndrome. Sometimes they package it into a pill. Yeah. So you can read more about this. This is actually helping people. So yes, we've proven that the microbiota does in fact affect a lot of us physiologically. Okay. Yeah, so thank you. This is one of the sequencing machines that we have these days. It's just as big as thumb drive. So this is the mean ion for Oxford nanopore, right? Yeah.
So
This is this device is called a mean ion, so it can be plugged into a laptop and you can perform DNA sequencing. Right? Yeah, it's from a company called Oxford Nanopore. Oxford Nanopore. It doesn't cost too much. I think less than 10,000 ringgit, maybe around 10,000 ringgit.
but
You need to buy the cartridge.
yeah, I need to buy the cartridge. That one very expensive.
Yes, there is some kind of cartridge which you need to change when you want to sequence a new sample. Maybe not too expensive. I think still
2,000.
still reasonable for researchers.
But there's no limitation on these devices, right? Because
Yeah.
there's not like large samples.
Yeah, of course. But the advantage is that now you can
Convenient.
go to places far away from labs and perform research, like deep in the jungle or in the desert or maybe in remote villages where there is no hospitals or that. Okay. Any questions so far before we go to the next part? Yeah.
I think 3 minutes or like.
Yeah, sure. Okay, so we'll take a short break and then we'll come back. The attendance you can sign wherever on the sheet. You can click online, yeah.
I tried.
It doesn't work.
Did you?
Yeah.
I couldn't also.
Not for today.
Not for today.
Previously it worked.
Yes.
Oh, okay. I will check again after this. You can also sign on the sheet of paper.
study.
So Kavita say, for urine samples, we determine if the pathogen is clinically significant based on the bacterial colony count. If less than 10 to the power of 4, we conclude the bacteria is insignificant, and sequencing able to perform on that. Yeah, so sequencing is is a non-biased technique. So in the sense that metagenomic sequencing, it samples everything or it sequences to find everything. Yeah, so non-biased means that it doesn't particularly look for any target, unlike RT-PCR, so reverse transcription PCR or PCR methods whereby they have certain targets of a panel of commonly occurring diseases. So in the case of metagenomic sequencing, you are sequencing everything and identifying everything, and then from there you will rule out if the bacteria is a pathogen or non-pathogen. So most likely if there is something which is not not commonly found, for example in urine, then you can suspect that maybe it is certain pathogen related to UTI or some other diseases. But technically urine is supposed to be almost sterile, right? Because it contains urea, and urea breaks down or denatures a lot of the proteins. Unless you have an infection in the kidney, or compared to sequencing is non-biased, so they will just try to identify all the possible microbes or even virus inside the sample. More frequently, then the counts indicate that there is a higher number of that particular sequence, which means it could be that the organism is more in quantity. So maybe it's semi-quantitative. So the next part is basically we're going to do some class activity. Okay. So during Father's Day this year, so I shared this with my father in the WhatsApp, that in order for us to be born, of course we need two parents. But in a short, actually you count, right? So your parents is the first generation, like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So in just 11 generations, you have 2,000 ancestors in order for us to be here today. And this is only in about the span of about 400 years. So if you take another few hundred more years, then you can go back even more generations. And I think in certain countries like China, Korea, Japan, they keep a very good record of their ancestral history, and then you can sort of trace, how many generations your ancestors here. And some Malaysian Chinese, some Chinese Malaysians, they can actually trace back to which family they come from. So, and also it depends on whoever you are, depends on these people. So what I'm trying to say is that are we who we really are, or are we the result of all this genetic material. Right. So you have no control of who you are essentially, because you are just the product of all this that allows us to be here. So you have something that is not to our liking, so it doesn't mean that it is our fault. It's just what we inherited from all these ancestors. Okay, and we can't change that, of course. So speaking of inheritance, in May this year, so there is a headline in the local news that there is a baby with a rare genetic disorder. Okay. So could you open up this, your search on your laptops, this particular news? So the news. Do you find it already? Yeah. You can read about the news first. Everybody got it already?
Yeah.
Yeah. Okay, so for the next 50 minutes or 1 hour, you will again be split into groups. In groups of, because Tabitha is not here, but you can still discuss with her online if you set up WebEx or through WhatsApp. So let's break into groups of 3 or 4. So 3 will be better, or 4?
3.
3.
No, 5 would be better.
Yeah, 5 better. Yeah. 2.
I have 10 people.
10 people, but we have 12.
Oh yeah, 12 more. Then 3?
Okay, so we have break
4.
groups of 3.
3 or 4.
Okay. So before we continue, alright, so let's look at what is required for a good case study. So have you used Google Bard before?
Google Bard.
Bard, B-A-R-D. So Bard is an AI assistant where you can ask any question and we can help you to summarize things.
I don't check.
Yeah, it's something like ChatGPT, but it's by Google. Okay, so I asked Google Bard what are the principles of a good case study. and he gave me these results. So a good case study is a compelling narrative that delves into a specific situation or problem, examining the actions taken and the outcomes achieved. So it serves as a valuable tool for understanding real-world applications and drawing insights from practical experiences. So here are the key principles of a good case study. Clearly define problem and context, okay. Engaging storytelling, structured approach, has emphasis on action and impact, detailed and credible data, means that there is proof, right? Whether it's quantitative or qualitative. Relevance to the target audience, balanced perspective, insights and learnings, and call to action. So these are some of the things that is sort of necessary for a good case study. Of course, I'm not suggesting that you follow exactly all of this, okay? But in the next 14 minutes until we finish, so let's break the groups and then we are going to discuss in your group. Disorder in that particular news story. Okay. How can it be treated? So there are two things. One is the genetic disorder, what is the background behind it, and how can it be treated? How can we take action to either treat person with already this disorder, or we can take a preventive action to prevent or to diagnose somebody. Any questions or understand what I'm trying to say?
So we have to identify what is the disorder,
Yeah.
how it can be treated.
How it can be treated, or
Prevent
or prevent it even before the person has it. Can it be prevented, for example?
Okay.
Yeah.
So these two.
Yeah.
So we have to present it on.
No, not present it on 14. So for the 14 one, you have to present a case study from a published article, like what I've shown you for the one for the boy just now. So the one that I showed you earlier, that is for the 14.
The boy.
But for the discussion now, for this one, is an exercise for us to understand about genetics, okay, rare diseases. How can it be treated? Do they use some kind of bioinformatics tool to diagnose or even to treat that person? So there are many aspects that you can explore.
Mm-hmm.
So these three groups, I think by the end of today, we will just give a brief summary of what we have discovered, what we have discussed within your groups.
So if you present it in the PowerPoint or
Just maybe one slide PowerPoint. Yeah.
one slide PowerPoint?
Yeah.
One slide.
Or how many slides do you think?
Prefer no PowerPoint.
Just for today's one, yeah. So we want to summarize what we discussed. Even without PowerPoint is also fine. If you can just speak the main points and share it very well.
So break into three.
Yeah? Yeah, you can split into three groups, or groups of three or groups of four. That is fine. So today we have 11 person because Kavita is online.
How to do it?
So if it's a report in the state study, is this
No. So for now, but for your own.
these are very long. So we have to talk about the state study, genetic disorders, the principle of how we we can say it just opens out what they can also need. Okay, expansion.
Purpose, we are so we are
Easier to sit here. So, Kavita, you can explore it on your own also. Kavita joining online. Yeah, Kavita can also explore on offline. For 1-3.
Sorry? Can you read the article? The article, what is disease? No, it's fine. For the sounds, I know. Because it doesn't have. So it sounds. So I haven't read it.
What? Oh. Mình sỏi thật quá. this? SMA. Hmm. 좀 씁시가요. 운동神经元的存 What's this? Wow. 베이징. 预防治。
This then I would explain you guys this from as a setup. I said to you guys this coaching out the I the same feedback on is that the same feedback on the form which is smart. So this assembly time is what I show you this is for the ruling improvement also known as spiral. Spiral means that it usually happens in the spiral. But then usually so actually some fitness spine muscle for means is case is the next. So it's very fast. No, that provided is using so, what does this so it's a genetic disorder we carry error system in translate the gene number because of the errors and the fault so it's a因为整个有一个不好的okay so you have to listen from you have to the condition is in order for you this type of it need to have parents mother and father at the same time. So this means that father and father contains or liquids together so they can distance happen. Okay, the father so there's the so anything so usually we have a carrier because but the carrier doesn't affect the first explanation you need to have those two important so we express at the same time until you can get distance when you have healthy gene. One healthy gene that doesn't affect if I'm the losing carrier, but that doesn't affect carrier means that you actually have a chance to get affected. This should be type of okay, so this portion is actually so as mentioned it's 4 months. 4 months really diagnosed. So 6 months after 6 months. So we have difficulty so it's because so where does it affect? It's a spinal muscle. 我多年是那个三个不一样的setting，我多年是那个 effector 运动我多年，so 它会影响你的这个运动神经元素。
它是不是一种让运动神经元就是相当于让它没有实力的东西，没一种条件。哦，我还没有这个使用。我你们要不要中文的？
Okay. So actually, the motor neuron is SNN protein. It's called SNN protein. The is called SNN, or it's so slightly big. So without this SNN protein, the motor neuron doesn't produce like protein. I'm not sure what protein it is and what's the function from like the general uses. I just know that this SNN protein is crucial for the motor neuron device, but I'm not sure what it means. Okay. So 你要这个也是比这个大孩子才能讲，你们知道为什么他学是什么？他经常，然后就是看他好，都还有呢个我是想带来，来外国without this and not this and Do you read science or do you so what is in science? What really study okay, so this one, okay, I don't know. In this article, I haven't read some. Okay, so the content properties is as a one protein. So this is an example. If you take away for example, so it's called fully functional. So it will survive because the protein is in core shape. Right. So all right. So that's one. Photo you want to generate.
Oh?
So, everyone, yeah. So, we need to know how we diagnose this. So, in this case, usually when you're having a muscular dystrophy,
哎，接着看。
So, I think faster biopsy is the most accurate. But this usually relate to some other disease like state. So you can run a genetic test. You can do a EMG. EMG is actually the test that you feel nerves. And this muscle virus is a procedure used, like I said, to the lab. Isolate. It's all ways to use. So, it can be maybe that during pregnancy. Yeah, how do you
诺西纳生
So Jeff, one sandwich harmony XIY is for sure.
Wow, that's very helpful and informative. Yes. So, okay, let's say if your period you can
单剂量基因替代
all the same, like make sure you choice. This is not safe for mistakes. You actually run for these tests. I think this is very common in most industries. You remember? Most industries, I mean, it's very common. This probably is yeah, I think also is very common in use. So you answer.
没什么用。
一个是阳台输出啊，一个是那个有个老师啊，是在那就CDF 去 So yeah, okay, so throughout the reading I found there's actually two ways of this. Two ways to do this. One is you need to take the medication, and another one is use the gene therapy. So for medication, usually it's only the only applies each diagnosis two of them, yeah, drugs. So FDA approved drugs of so you can use that one. Okay. So your slides is strength. So how to take this drug? They are either like injection with or it's not for another different option is it is blood or just so we can take that something is daily so what if so diagnostic children in this case because this article says that one therapy which is very costly, right? So this method gene therapy because using travelers. So this is not as smart. So we not is actually virus stopping healthy, and then you deliver by using so this is the therapy replace the missing of this is the real content. I think it's my people inspired this therapy actually is delivering the gene itself by using antivirus. Science the cell so you use the complete functional SMN. Remember this is the SMN 1364 has the reason in order to explain the number of hence the traits. So this it comes preventive have this reason but you can not or you may also use IBM. So IBM actually very smooth. So although I'm sure you should have the essence. So IBM is actually such yes. So anyone follow the slides? Make a slide. So the general okay, like that. All the over time.
诺健生。
What the
做个呢？做个森吧，做个森。
So you're fine to finish up.
Doctor, email the Gmail.
Yeah, sure. Thanks, Captain. Any comments? Yeah. So you'll present and share with everyone. Kavita, you can also present from the WebEx.
Okay, do I have to share the screen or something?
Yeah, when it comes to your turn, then you can share the screen.
Okay, noted, doctor.
Okay, so let's get going. All right, everybody, back to your seats. Don't worry. Yeah, we'll just present whatever you've already discussed so far. But, yeah, don't worry. Yeah, doesn't have to be perfect.
So ugly.
What?
Kavita can also share your slide or your document. You can click the share button at the bottom. Okay, so can we start already?
Yes.
Yeah.
Not yet, but yes. No. Very ugly. Eh?
Well, there's no points on it. It's just for learning together. Yeah. Okay, so everybody get back to your place, and then Kavita will present first. Yeah, she's ready. Right?
Wow.
So we want to pay full attention to her. Okay.
Hi.
All right. Okay, so Kavita, you can start.
Okay, sure. Can you see my slide?
Yes.
So the definition is neuromuscular diseases affect the function of muscles due to problems with the nerves and muscles in your body. The most common sign of these diseases is muscle weakness. So what is spinal muscular atrophy? It is a genetic condition that makes the muscles weaker and causes problems with movement. The type 1 is the most common and severe form of SMA, called Wernicke-Hoffmann disease or infantile onset SMA. Children with type 1 have limited movement, can't sit without support, and have trouble breathing, feeding, and swallowing. Symptoms begin at birth or within the first 6 months of life. So it is caused by a defect, mutation in the SMN1 gene on chromosome 5. As you can see the slide, we have the SMN1, SMN2. During the splicing and translation, the normal SMN protein levels and low SMN protein levels differ. So this is the diagnosis. So with the, hold on, I can't see this. Okay. Once the diagnosis is confirmed, sorry, did you get that? Hello?
Yeah. Okay.
Yep.
You can continue.
Yeah, so when it's SMN1 gene detection test, homogeneous SMN1 deletion detected, 5q SMA diagnosis confirmed. If there's a homogeneous SMN1 deletion, non-detected. So now we go to clinical examination, EMG, NCS, CK. The typical symptoms, neurogenic EMG or normal CK. Then proceed with SMN1 copy count. Then the 2 SMN1 copies, the other one is 1 SMN1 copy. This is considered other motor neuron diseases, 2 SMN1 copies. If the 1 SMN1 copy, it is then gene sequence, and no SMN1 mutation found, the SMA remain unconfirmed. But if the SMN1 mutation found, then the 5q SMA diagnosis confirmed. So once it is confirmed, the treatment starts. Combinatorial treatment enhancing SMN2 gene expression and inducing SMN2 splice correction. So this is the gene replacement. Other than that, a conservative treatment is a multidisciplinary approach, key for the management of individuals with SMA. In SMA, it is important to monitor the various aspects that are known to be involved in disease progression and, when possible, provide anticipatory care. The MDT can consist of acute care clinicians, consultant neurologists, gastroenterologists, neuromuscular care specialists, nutritionists, occupational therapists, orthopedic consultants, physiotherapists, physicians, psychologists, pulmonologists, speech-language pathologists. Apart from that, we can also see how the SMN gene inserted into viral vector. This is the gene. It is from the viral DNA. The gene encapsulated in this virus that is not integrated into the human genome. This SCAAV9 carrying the SMN gene is delivered into the targeted cells or tissues of patient via injection. So SMN gene is introduced to target cells or tissues from virus SCAAV9. And then the SMN expression is restored to normal level in cells which have the viral genome. Another pathological treatment, pharmacological treatment is risdiplasm. Mechanism of action is this risdiplasm binds two sites in SMN2 pre-mRNA, 5S of intron 7, ESE2 in exon 7. And then binding to the 5SS of intron 7 improves recognition by U1SNRNP. U1SNRNP promotes inclusion of exon 7. The unique specificity of binding two sites increases levels of full-length mRNA in protein while reducing the impact of splicing of other pre-mRNA. Thank you.
Wow. Okay, thank you, Kavita. Very. Yeah. Any questions?
Very informative and complete.
So we can go back already?
Yes!
Thank you, Kavita.
Thank you. Our slide is rubbish compared to yours.
Yes, totally rubbish.
Rubbish
than you and you.
So we cover different aspects of it. So there's no right or wrong because we want to learn about this disease. Learn from each other. Okay, so who's next?
Oh, yeah, go next.
Yeah. Okay.
You go first. Well, we don't have the slide.
You can just present from here because Kavita will see the screen from here.
Oh.
Yeah.
So maybe
Admit. No. How to join the WebEx?
so let's send the link.
Yeah.
That's the link. Sorry. Hey, yeah.
Okay, next will be from this group together with Leo.
Okay, good. So our slide was not so as complete as Kavita. So okay, so right. So from the articles, it states that children is diagnosed with this SMA type 1, actually there's 4 types of SMA. So this is type 1, which is the most serious one. So SMA type 1, also known as spinal muscular atrophy, is a genetic disorder where the patient has inherited 2 faulty SMN1 genes from the parents. So you need to have 2 faulty genes, one for the father, one for the mother. Because it's one's father and mother. You need to have these 2 faulty genes together, only you will develop this disease. Okay. So if you ask me what if you have one, so that will make you a carrier instead of infected person patient. So these SM1 genes encode for this SMN protein, which essentially is the survival function of motor neuron. So as what Kavita explained just now, it is used during the splicing process of these motor neurons. So when this you have this faulty SM1 genes, SM1, SMN protein, the motor neurons couldn't function, and this caused the prevention of signaling from brain to muscles, right? Because you don't have motor neuron, your signal won't able to direct it to your muscles. So in long term, muscle will progressively weaker and shrink, and this condition is known as muscle atrophy. So it happens in your spinal. So that's why it's known as spinal muscular atrophy. So this is quite a simple diagram I like to use to explain. So I have a mutation SM1 gene. So this deficiency in the SM protein leads to splicing effect in motor neurons. When you have a loss of motor neurons, it's not like loss, the motor neuron doesn't function properly. In the anterior horns of the spinal cord, it prevents signaling between the brain and skeletal muscle, and hence your progressively muscle weakness and atrophy. Right. So then for diagnosis for this, right? Okay. So as usual, you have like 4 different methods for you to diagnose whether you have this disease or not. So blood test is a testing for creatine kinase. This creatine kinase is released by the faulty motor neurons. Usually, this enzyme will be released by the faulty neurons. So when in the blood test they detect a high level of this enzyme, it could lead to the result of you getting these diseases. But it could be other diseases as well. So blood test was not so accurate. So genetic test will be slightly more accurate where you have a newborn screening. So you check the particular muscles when doing a genetic test. Nerve conduction test is EMG, also is explained by Kavita just now. We test the activities. After 2 years old. I'm not sure of that, but that's what I read. So medication, you have this Evrysdi and Spinraza. Evrysdi is referring to Ridaspar. This drug is called Ridaspar. I'm not showing you, but it's okay. And this Spinraza is actually a drug called neurosteroid. So another therapy for this is you can go through a gene therapy. So let's say your children is less than 2 years old. Yes, you can go for this gene therapy. It's an IV of this onatimogene abrarotioid. This is the drug's name. So Vesma, the name, but it's actually a copy of a healthy SMN2 genes that will help you to produce a functional SMN protein. Okay, it's delivered by AAV. AAV stands for adenovirus, if I'm not mistaken, into your targeted muscle cells. Okay, so this is how you can do a gene therapy. And prevention for this, let's say you want to prevent it, so you can go through IVF, where the doctor is already genetically modify everything completely for you. Not genetically modify, but to make sure to insert this gene, not genetically modified, actually to insert this gene into the test tube babies. And
But that is for screening only, right? Screen. Yeah. They cannot modify because
not ethical. Sorry, not genetically modified. It's actually you
get screen
Screen?
idea.
yeah. So this is the picture of a children who's suffering from this disease. And doctor is smiling at me. I don't know why. Maybe he's evil. So yeah, that's all from us. Thank you very much.
Thank you.
All right.
So when you present to everyone, not to me, because I'm also learning from all of you. So who wants to go next? Ian. Okay. Ian. Okay.
So, speaking words.
Yeah.
Yeah, the baby. All of us will be.
Big. Yeah, sure. Yeah. But you have to speak louder so that Kavita can hear. It's okay. You can pick up. All right. Yeah. Okay.
SMS except autosomal recessive disorder. Autosomal recessive disorder is a disorder when there's two abnormal copies of the gene, then it's an autosomal recessive. So the deletion occurs at SMN1, survival motor neuron 1 gene at the location 5Q13. So basically there are 5 types of this condition. Type 0 is extremely rare, and type 1 is severe, and 3 and 4 can then survive, can have a normal lifespan. And the symptoms usually appear at the first 6 months of life. Okay, I'm going to talk about the residences or mechanism. So, like Subhana mentioned, this SMN1 disorder is caused by the mutation in SMN1 gene. So, the main function of SMN1 gene is to include a survival motor neuron. But then in this case, the exon 7 of SMN1 gene is deleted, due to which survival motor neuron cannot be produced. As a result, the motor neuron starts to die, and then, as we all know, motor neuron is a signal transmitter. So there will be a signal to the brain and the muscles. As a result, the muscles cannot contract and muscles can bleed and waste into the spinal muscular
Okay, so I'm Ian. So I'll be talking about the treatments. So there are 2 types of treatments. One is the supportive treatments. Okay, so supportive treatments, which means that the patient have to take this treatment for the whole lifespan of his or her life. Okay, so there are 2 supportive treatments, Mirana and this. So these 2 supportive treatments are a long-term supportive drugs, which fix some splicing errors in the genes of SMN1 or 2. Okay, so there's another treatment which is the gene therapy. So this gene therapy is only taken once in your lifetime, but not like few times in the life. So once in a lifetime only. So it's already approved and licensed by FDA. Then the drug is known as the Zolgensma. So this drug is contained of SMN1 gene, and added into the adeno-associated vector 9. So this adeno-associated vector 9 will bring the SMN1 genes across the blood-brain barrier, so that these genes is included into the cells, so that the cell transcription factors can transcribe the genes of the SMN1, so that it will produce a functional SMN1 protein for the patient, so that the muscular functions becomes normal. Okay, that's all.
Yes.
Okay. The last part about our presentation is all about the prevention. So as we know, like this is the genetic disorder, so we can do like there is no any option for us to do the any prevention as any other infectious diseases. So we have to look for the genetic level of the diseases. Firstly, like as my friend all mentioned about that it's had evolution about the exon 7 of SMN1 gene. So if we have like the parental, firstly we have to do they have to do the parental screening. If we have the parent with both of the parent they carry for that gene, so it means like 25% of their offspring they will produce this kind of the genetic disorder. So this is the first line of the prevention. The second one, like right now they are working on that thing, because like from the parental screening, sometimes like the parent they cannot decide to have their kids in their life. But from the second line of prevention, they have pre-implantation genetic diagnosis. This is happened through the IVF. So I think they mentioned about that thing. Also, there is another way they are working inside the lab. They call it CRISPR-Cas9 system. They are working on that system to modify the genetic sequence of the offspring to prevent that disease to happen. Thank you.
Thank you.
So at the end, genetically modified baby is still
Maybe it's the future,
possible.
50 years from now.
But we have to sort out the ethical issue and everything.
Yeah, but as a parent, who wouldn't want to have healthy offspring?
Super baby.
Yeah.
A big already, right?
But it's not
Even it's
approved.
not fault, but it's not
It's already
But it's not perfect yet. Yeah, technology is not perfect because there's a lot of off-target and off-sites. I think in China, the scientists say that they modified the babies, but
It's not approved, right?
that,
Yeah.
it's not approved. Yeah, I think it's not. I think the final update was that it's not perfect. Yeah. Okay, and the last group, Halin will share.
What is the genetic disorder? The spinal molecular atrophy type 1. I read that there are 4 types, but the first type is the most severe one. And as previously mentioned, the infant cannot sit down, swallow, or feed. And this is considered a severe one. And also, I read that it's a recessive gene, so it means that both parents need to have these genes for the child to have this disease. Both of the parents should have this gene. People with SMA are either missing part of SMN1 gene or have changed or mutated genes. A healthy SMN1 gene produces SMN protein. Its motor neurons need this protein to function properly. Therefore, type 1 is the most common and severe form of SMA. It's sometimes called Werdling-Hoffmann disease or infantile onset SMA. Children with type 1 have limited movement and can't sit down without support, have trouble feeding, breathing, and swallowing. Symptoms begin at birth or within the first 6 months of life. Many many children with type 1 do not live past the age 2. How can it be treated or prevented? We also read that there are 2 treatments.
One of it is the disease modifying therapy and the other one is gene replacement therapy. So we can refer from the article just now. Actually what they are using now is gene replacement therapy. This is
Zolgensma, which is one of the gene therapy injection to prevent further motor neurons and muscle degeneration. And this is, we found this also from the story of that child. So what is Zolgensma? How does it work? It's made up of new working SMN genes that is placed inside the delivery vehicle called a vector. This is a virus that's called associated adenovirus 9, and what they do is that they first will take out the genetic informations of the virus, and they will insert inside this gene, the SMN gene, and therefore this travels throughout the body and delivers the new working gene to motor neuron cells. By replacing the function of SMN1 gene, it will enable the production of SMN protein and helps preserve essential muscle function. Motor neuron cell that would have died without treatment can survive and be maintained, stopping the progression of SMA. After a single infusion, replaces the function of the SMN1 gene to help the body produce SMN protein.
So actually we talk about what's how to prevent, as mentioned by previous group, that actually is I think the words prevent is not that so appropriate to use in these terms, but actually before we have decided to have a baby, actually you can choose to get a simple blood test. Actually it's like prenatal screening, or even before that. So that's why after that we can decide if we really detected that we have those genes, maybe you can decide what is the next we want to choose. So if for the prenatal screenings, we can decide use a chorionic cell sampling at the 10 to 14 weeks, aminosynthesis at 16 to 20 weeks. So by using this prenatal screening, we can help to identify this genetic disorder early. So,
yeah. Thank you everyone for sharing what you have learned. So this is, I think, throughout your next month course, we'll have a lot of case studies and presentations like this. But the purpose is for us to share and also to learn from each other, because that way then we can reinforce. And I think you can see from just now there are sort of different opinions or different ways of sharing, even though it's the same knowledge, like still SMN and there are 4 types, but we share in a different way. So that helps us to remember and which are not in some presentations. Okay. Sometimes too many details can also be not good, because then it is not suitable, or that we do not need certain types of information at this stage. Right? So you have to decide as well what is required or what is enough just for sharing. Okay. And okay, so if you want to learn more about SMN, you can also go to Uniprot. Okay, so I hope you will use these databases more. Although we learn about the disease from the phenotypic or the background about the disease, but also from more molecular level. Okay, for example, if you search the keyword SMN1, okay, which you have found from many articles. If you search in Uniprot, you will get a list of different organisms with SMN1, and the one we're interested in is Homo sapiens, right, human. It has a gold star, which means it has been reviewed, so the information can be much more verified. So just now we can see that nobody shared about the sequence. No, sequence is not important because we cannot understand the sequence, but maybe you want to say that, okay, this is quite a small protein. Of course, small is relative because it's only 31 kilodalton, and at this stage, perhaps from your experience, small and big is very difficult to judge, right? Yeah, but 30 kilodalton is considered a not very big protein, so it's considered as maybe mid-size to me. Yeah, between small to mid-size for me. You can also look at its structure. It also has a very kind of simple structure, which is a barrel. The barrel is where you have these chips, and then there's a form. Okay, you explore more on these coming topics. You can find also information from Uniprot about the disease, and this is coming from a more scientific background because they will cite the publications and so on that summarizes this. Okay, and if you're interested in the scientific details, you can also check the publications through the links inside Uniprot itself. So Uniprot is very useful, especially when it comes to the scientific information at the genetic and protein level, okay, and especially in this mixed mode program. Another one is from NCBI itself. So NCBI is a database not just on gene bank only, or the gene sequences, but you can also find information from textbooks such as this one in the NCBI bookshelf about SMA. Okay, so it'll tell you about the clinical characteristics, diagnostics, how to. But it is here, and information in NCBI or NIH is much more. I mean, for me, it's much more reviewed, or at least some human being has checked the information, rather than from some websites like blogs and so on. Okay, so you have to also be careful of the sources that you use when you are presenting your information in this course. And the last one is that this is also another interesting story about muscle mutation in the muscle gene. Okay, so if the mutation in the same gene but at a different position can lead to different outcomes. So this particular Olympiad athlete, okay, she has the mutation that leads to naturally a very muscular outcome. She doesn't have to exercise a lot, but she's already very muscular. Yeah, she has low fat. But in the same gene, if it's a different mutation, then it can cause again some kind of muscle atrophy. Yeah, this person, this lady, cannot walk. So this is a very nice article because it tells the whole story of how this lady helped to discover the mutation. So she's not a scientist, but because she was affected by this when she was a child, so she went to find the answer of why she's like this. And finally, one scientist allowed her to join his lab and to help out in some basic research. Okay, and then from there she managed to find out that okay, this person also has a mutation in the same gene, but it's a different mutation which leads to different outcomes. Yeah, so this is quite a very interesting story that also helps you to understand about science and genetics and science is not as straightforward, and we are all still learning about many things. Right? So none of us are perfect because all of us carry some small mutations that we do not know, although we function as we are, but still those small mutations may sometimes only appear like later in life, some is early in life. We'll meet again on the 14th of December. Okay, so you understand the assignment, right? So the assignment is pick one research article related to that it shows the use of bioinformatics
In diagnostic and
yeah, it can be anything.
so application in
Application of bioinformatics.
Application of
Doesn't have to be bacterial disease or infection. It can be maybe cancer or like this. Human, right? Not medically related. Unless you can show that the food then relates to a medical condition. Okay? Yeah, if you have any questions, you can feel free to email me.
Sure. So where will be the presenting?
At 6?
Yeah, at 6, right? Yeah. You can
In the
start.
class?
Yes?
In the class?
Yeah, in the class on the 14th. Yeah. If you cannot join for some reason, then you can present online.
I think I will have reason.
Okay, that's all. Yeah, goodbye, Kavita. Hope you recover better.
Thank you, doctor. But there's no attendance link for today.
Yeah, I will set up afterwards. Yeah, there's now the donations. Yeah.
Thank you.
Okay, thank you. Selamat malam. Good night.
Selamat malam. Selamat tidur, selamat makan.
Terima kasih.
Iya.
They won't allow you to do so. Because maximum is 3 cents.
我打电话也不回。
我给他打电话也不回。
蹲坑去了。拜拜。
Hello。
要去哪里？
你在哪里？要不要去吃饭？你上次不是跟我说你要吃炸鸡吗？你要去吃啊，带你去吃好吃的炸鸡。炸鸡？啊？对，我在门口。带你去吃好吃的炸鸡。我跟你说，这是马来西亚口音炸鸡。炸弹还是炸弹？
炸弹我当然是。
炸。
楼上。
他在楼上。在日落洞。不会啊。
日落洞吗？
日落洞。
我知道，我看过地方。要是应该是那个厕所。
不用吧？不要带他们去吃。
我感觉我可能撑不了1个小时。啊。天哪！我上厕所，他帮我拿着。我赶紧去。你放多少？放多少？我一会儿过来。看这个红包肉吗？
每天都没有做这个东西，这样子的吗？我是叛逆的小孩。今天不知道为什么马币跌得很厉害，这几天马币都跌得很厉害啊。今天忽然一下。
我用支付宝它都是1.53。
现在兑美元好像是4.7，4.8，1.525，今天4.8了吧？兑美元。1块美元兑马币4.8。再见。发微信我回到家。今天兑美元多少了？我看人民币是涨那边。人民币倒是有涨，兑美元吗？那是人民币涨了，不是美元涨了。对，我是说人民币兑美元多少？多少？兑美元兑人民币是吗？7.3，保持了，没有涨，没有跌。应该4.7了，我记得4.8喽，4.8今天4.8了。一直在跌，不知道为什么。这2天跌得很厉害，不知道为什么。原来是以色列战争那里跟那边，然后就来马来，然后就在讲马来赛，是美国的下地不是吗？你要去吃炸鸡吗？
走啊 ！Go go go！ 哎呦，哎，找个人，人呢？他走了。他不吃。你吃吗？走了。那我一个人吃干嘛？
不吃啊。大哥，是不是可以吧？
哇。
上车。
不是，
走过去吗？还是上车啊？
走不动啊，走不动就。你坐前面。 过了，味道没有什么差别，然后我就不想吃了。他们都是一个味的，所有的是原味还是吃辣味？我一般吃辣味。你不要吃原味，吃辣味好不好？啊？这里辣味比较，那个皮比较脆。哦，为什么？我也不知道。那辣味的基底是原味，辣。而且是啊，发现它跟就是我平常吃的没有什么差别。然后对原味的在哪里都一样。哦。但它没有那么甜辣酱，我看你们那其他酱也是。这也可能讲是给白痴吧，你那也是白痴。我选的是这个不可以选。你那可能是给可乐。对。我这些可能是给白痴。对啊，一个是可乐，一个白色，它是可乐跟可乐。我们这个是都可乐，也可能这样子给白色。中国的肯德基我没吃过点。但是华莱士的话好像都可以吃，不过长期好像没有买。那里好吃，但我买的。我一般是我们本地的东西啊。是它年老年，它味道跟日本差不多。现在在嘉义不在我们这边，如果跟他去的话，可能是同一家。那啥店？他中午我问他的时候，他说不想吃啥了。主要是看 CP 啊，不对，主要是看谁啊。跟一个帅哥去，好啊，我吃啊，我最喜欢吃炸鸡了，怎么？啊，对不对？我也是这样，选择性喜欢。来了来了。是德语是这样，看菜人下菜了，真的是下菜。那我就觉得很下饭，是吧？下饭是吧？对，真的下饭。那我感觉，我现在拿3天了，再过2天再换。换这样干嘛？啊？这个继续的时候换啊，后面慢慢换。我先换一会儿，我得先换一会儿。明天去了，你觉得明天可以换吗？明天去哪里换？找个地方去。你是怎么换？我是去楼层换的。楼层怎么换？那有ATM机啊。哦，那门口就有啊。我以为你带什么钱过来直接换的。我以为你带美金还是带那个过来？我是就是跟舍友换了300，然后先用着，后来就拿。你是用当天的汇率来换来的吗？1.56。那好啊，今天好像是1.5。这个1.5来了。所以现在用支付宝扣掉。哦，好问题。又忘记了。啥呀？下回忘记了。对，交钱吗？对。啊，我记得，但是我知道我懒得说。那他们也是吗？好了。 他去的不多，这是摩托车的位置。摩托车的这可以停，好像不太可以。他这个不是停到摩托车位上，那对吧？那就停吧。这样他是要走。对啊，是要走。然后朝那边不能停。行吧，他们都要了。将停喽。我觉得应该没有人管吧。主要是你这也是在人家饭店门口。Ah? Tak ada. Ini tak ada khas wah. Ini khas untuk kedai ni. Ini ada khas kah? Ini ada khas kah? Ini motor parkingnya. Oh, motor parking ni. Oh, kalau makan sini kita boleh parking lah. Makan boleh lah kita. Oh, okey.